Hello, everyone. Welcome to today's live broadcast, Friends with Brain Benefits, the impact of gut microbiota on neurodevelopment and behavior. I am Brenda Kelly Kim of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars, advancing scientific collaboration and learning. Before we start, there are a few instructions. We want to hear from you during this interactive broadcast, so please ask questions or leave us a comment. Answers are always welcome too. You can do this by hitting the green Q&A button at the lower left of the presentation window, typing in your comments and questions. We'll try to get to as many as we can, and we'll follow up with you if we don't have time today. Want a better look? You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you can't hear or see this presentation properly, please let us know by clicking on the support button on the top right or use the Q&A button. We'll try to make sure we resolve any issues. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the process to obtain your credits. Now let's get right to today's presenter. We are proud to welcome Dr. John Cryan. John Cryan, Professor and Chair, Department of Anatomy and Neuroscience, University College Cork, Ireland. He received a BS Science and PhD from the National University of Ireland, Galway, Ireland. He was a visiting fellow at the Department of Psychiatry, University of Melbourne, Australia, which was followed by postdoctoral fellowships at the University of Pennsylvania, Philadelphia, and the Scripps Research Institute, La Jolla, California. He spent 4 years at the Novartis Institutes for Biomedical Research in Basel, Switzerland, as a lab head, behavioral pharmacology, prior to joining University College Cork in 2005, where he was appointed a full professor in 2011. Professor Cryan has an H-index of 68, having published over 300 peer-reviewed articles. He was selected as a highly cited researcher by Thomson Reuters. He has edited 3 books. He is the senior editor of Neuropharmacology and Nutritional Neuroscience and an editor of the British Journal of Pharmacology. He is best known for his work on animal models of stress-related disorders and their pharmacological treatment. More recently, his team has been instrumental in demonstrating a role for the gut microbiome in regulating brain and behavior. I'll now turn it over to Dr. Cryan for his presentation. Cryan, for his presentation.
Good morning, everyone. Thank you, Brenda, for that kind invitation. It's really an honor to speak among this great number of people on this LabRoots symposium. And the goal of this is that you can do things without leaving your home. Unfortunately, I've left my home. I'm doing it from a hotel in Germany, so I hope it all works out okay. And again, this is St. Patrick's Day, so I'd like to wish everyone a happy St. Patrick's Day, wherever you are celebrating in the world. And I'd also like to welcome those people who are watching this back on demand, and you're very welcome as well. So what I'm going to talk about is the journey that we've been on in our lab over the last number of years, trying to really understand how your gut could influence your brain, and the implications that that might have for particularly stress-related psychiatric disorders. It's really become a paradigm shift in neuroscience to start thinking about anything below the neck. And so over the next 45 minutes or so, I hope that I can persuade you that there is a lot of value to trying to understand this complex interaction. And I work in a medical school, and in medicine in general, the fields of microbiology and neuroscience rarely talk to each other. There have been some exceptions going back to the time of syphilis and neurosyphilis, and more recently when we really appreciate the cognitive sequelae of the HIV virus and may have in patients that are surviving. But for the most part, these disciplines are quite distinct. And even the discipline of gastroenterology and neuroscience is another one which is quite distinct, even though we have what's known as a brain-gut axis. So I want to take you back. I want to take you back to rural Michigan in the 1800s, and there you have this U.S. Army surgeon called William Beaumont. And Beaumont was one of the prototypical clinician scientists. He was very interested in how the body worked, and we knew very little back then about how the digestive tract worked. And he had a patient called Alexis St. Martin, a Canadian fur trader who unfortunately got an abdomen wound caused by a gunshot. And Beaumont saved his life, but a fistula emerged, an opening in the side, and that allowed Beaumont to get really good insight into the physiology of the digestive tract. And he started performing experiments and writing about it by looking at the rate of digestion, the what was in the digestive juices, etc. And so he kept St. Martin as almost a human guinea pig for many years with this opening doing these experiments. And he noticed that if St. Martin became, understandably at times, irritable or angry, and who's to blame him, that it affected the rate of digestion, that your emotions changed what was going on in your gut. And this was subsequently taken up by many of the greats of modern physiology, from Pavlov to Cannon to Claude Bernard, and even Darwin has written about it in his classical text, The Expressions of the Emotions in Man and Animals. So we have this brain-gut axis, and with the advent of imaging studies in the 1980s, we began to really realize that this is bidirectional. Here is a MRI scan from Emmer and Myers group at UCLA showing that distension of the colon can cause activation state in key brain areas that we know are very important for regulating emotions, such as the anterior cingulate cortex. So we have this bidirectional brain-gut axis, and the concept of mind-body interactions really, has been very well appreciated in complementary medicine fields, but in traditional medicine, it has had less of an impact to date. And that is despite these well delineated modes of communication, including here, I'll point out the vagus nerve, because I'll come back to that a little later. And so it's still seen as a little bit out there that we should be studying this brain-gut axis in neuroscience. And my lab is particularly interested in stress and how stress affects the brain, and how stress, and particularly traumatic stress or early life stress, will have a long-term impact on all aspects of the body. And we're living in a stressful world, and everywhere we go, and so understanding the mechanisms of how stress is having such impact may help us to get better treatments where there's a clear need to have. And it's worth noting, as pointed by others, that stress will have different impacts depending on when across the lifespan it occurs. We're quite interested, as I mentioned, in stress in the perinatal period, but also stress in adolescence and stress in aging. And understanding what's going on in the brain during these key periods is also something that we're quite intrigued about, and how that will have different impacts on behavior and on physiology in general. And I really like this quote from Hans Selye, who's the father of stress research. He said that it's not stress that kills us, but it's our reaction to it. And so really understanding the neurobiological basis of why, if two people are on the roller coaster of life and they're subjected to the same stimuli, that one will be stress sensitive and the other will be stress susceptible. And what are the factors underlying this, not just the susceptibility, but a lot of more emphasis now is going on to understanding stress resilience. And we know that genetics plays a role. We're getting a lot more information about the epigenetic influences. But what if it's got to do with something in your gut? What if it's got to do with the microbes within your gut? And I'll show you over the next bit at a time, the information that we've got that helped us to come to that kind of conclusion and where we hope that would go overall. And, it's perhaps not even, we use the gut in our everyday language all the time. We have phrases like gut feelings or gut instincts. We have butterflies in our tummy to personify our emotions. And so maybe what we're seeing is that our emotions relevant to stress may be gated by what's going on directly in our gut. And we know that stress, as I mentioned, impacts the entire body. And I just wanted to point out that while it does have direct effects on key circuits within the brain that we're also fascinated about, but its impact on the immune system, and then therefore on the immune system's bidirectional communication with the brain, is very important to understand. Stress will also have effects directly, locally, in the gastrointestinal tract and changing lots of the physiology that's going on in the gut. And in particular, will have an impact on gut barrier function. And that barrier function is important for maintaining an appropriate immune response overall. Now, I'm a behavioral neuroscientist, and so one of the issues we have is trying to use animal models to translate what we can do in basic neuroscience into the clinic. And I like this cartoon in particular from Gary Larson. And not just because it is an Irish setter and it is St. Patrick's Day, but it is, it really encapsulates the difficulties that we have in behavioral pharmacology in trying to understand what's going on in other species. And we can't just put our mice or rats on a couch and ask them how their parents treated them in their early life. We require behavioral assays that are robust, reliable, and translatable to be able to make that leap forward. We also, and so there are, of course, distinct challenges in this, not least of all in terms of the anatomy of the rodent brain and the human brain, where there are distinct differences. Well, we can and do use rodents with a certain amount of confidence when we're looking into circuits that we know have been evolutionarily conserved, be it the reward pathways in the brain or the fear and anxiety circuits through the amygdala. And a number of years ago, together with my close colleagues, Ted Dinan and Siobhan O'Mahony in Cork, we were interested to see, well, what happens not just in the brain, but also across all the different systems in the body if we take rats and stress them early in life. And we use the maternal separation model that's been widely used by Paul Plotsky and Michael Meany and many others. And what we found was that when rats were separated from their moms for 3 hours a day from postnatal day 2 to 12, when they grew up, they developed a wide-scale change across different systems. They had increased abdominal pain. They had an exaggerated HPA axis response to stress. They had behavioral changes. They had a pro-inflammatory phenotype. When we looked at their guts, there were changes in permeability and in morphology in the colon. And so we were beginning to see that really to reinforce the whole body effects of stress, a stress that happened early in life. And these are now adult individual rats. Now, what we did, and there was almost a look-see experiment at the time because we had colleagues just down the hallway who were able to help us in this. We also looked at the composition of the microbes in the gut, the microbiome. And what we found, using very crude techniques at the time, was that there was a shrinking of the diversity of the microbiome in animals that had been stressed early in life. That there was a signature of their early life trauma that persisted into adulthood. And this was really intriguing and got us thinking, if stress can change the microbiome, is there any other way that we can understand this better? And more recently, we've gone even back further and looked at prenatal stress, so stressing moms. This is again in rats, and looking at the offspring's microbiome in adulthood. And we find remarkably that, again, there are persistent changes in the microbiome, and that we can correlate these changes with changes in stress-related responses, including changes in corticosterone and in the physiological breathing response, which hasn't been shown before. So long-lasting impacts of stress on the microbiome. This has recently been also shown in human studies from Holland, where they looked at moms that had high perceived stress during pregnancy. And again, the infants, when they looked at the infants' microbiome later on, they had a different microbiome, and they were able to correlate it with the stress in the mom. So again, this field is slowly moving into a more translational way, and that we're able to recapitulate some of the effects we've seen in animals in humans. And so it got us really exciting overall, that now we can see probably, for the first time, that really that stress is impacting the microbiome. And except, in science and in medicine, there are very few new ideas. And so if we look back at older literature from the 1970s, there's a very nice paper from Jared Tannock and Dwayne Savage, which showed that stress mice showed dramatic reductions in population of lactobacilli. It wasn't taken up by the field. There was some subsequent work by Mike Bailey and Chris Coe, but very little work had been done in that intervening 30 odd years or so. So we're now beginning to see that stress affects the microbiome. So what is this microbiome? And I guess that's why many of you are tuned in today to hear about the microbiome. Well, we are living in a microbial world, and that's an important thing to say, that when we look at our genetic material, we are 99% microbial. If you think about the amount of money we have invested as taxpayers around the world in human gene studies and the Human Genome Project, and that accounts for less than 1% of what our genetic makeup. It's quite humbling in general. If I was giving this talk some weeks ago, I would have told you that we were probably about 10 times more cells that are microbial than human. Again, showing the field is moving fast, and with technology, we're getting more and more information. And now that's been revised in a paper in Cell to that we're about 1.3 to 1 microbial. We're still more microbial, but just about. And every day as we shed bacteria when we go to the bathroom, and perhaps we are just becoming that little bit more human each time. And so that's an important factor to take into account. The other one that I want to reinforce is that the weight of our microbes is roughly about the same as our brain. So again, it's about 3 pounds. Again, it's quite humbling as a neuroscientist to think about it. And also, I need to tell you that we, and I'm guilty of this too, often just think about the microbiome as being bacteria, but also there's some really great data coming out now in relation to viruses, the virome, phages, fungi, etc. So we can't neglect that, and that we have co-evolved with these microorganisms to become what's known as the human holobiont. It is perhaps one of the biggest areas in all of medicine. It's making the cover of all of the glamour magazines. It's making the cover of the science literature, making the cover of the New York Times Magazine, the cover of The Economist. There must be money in it. It's creating a lot of innovation, a lot of startups coming in this field, and it's very exciting overall, I must say. So where does our microbiome come from? Where do we get our microbiome? Well, for the most part, we get our frontier bacteria from our moms as we are being delivered into the world through the birth canal. So it's another thing in psychiatry, unfortunately, we can blame our mothers for. And it was long thought that an individual in the womb was maintained sterile, but now we know that the placenta at least harbors a unique microbiome. And this has been also challenged evolutionary by Seth Bordenstein and others, the concept of this sterility. But for the most part, these early bacteria that play a key role in forming our immune system and laying down the colonization of the gut come from the birth canal and from the environment. And that is if we are born by normal delivery. But over a third of you and a third of the general population will be born by C-section. And if you're born by C-section, things are a little different. And work from Gloria Dominguez-Bellows group in New York and others have shown this quite dramatically, that there's a different microbiome present in infants born by C-section compared to vaginally born. And the health consequences of that are slowly only being resolved. We know that if C-section born infants are more likely to develop allergy, asthma, and immune-related disorders, but we're also now beginning to think, well, what about its function in relation to stress sensitivity or brain health in general? And studies are ongoing to tease that apart if there is any major effect. And if there was, I think we would know it, but about it by now. But it's also important from a public health point of view. In countries where you have skyrocketing C-section rates like Brazil or in China and in southern Italy, and real regional differences in obstetric practice, that people just need to be aware that there might be potential health consequences of that. And a lot of effort is now going into trying to come up with ways to be able to change the normal C-section born infant's microbiome to be more like that of a vaginally born through different, either through a feeding process or through a dietary means. Speaking of diet is another factor which will really influence the microbiome. And we know that kids that are fed with formula have a different microbiome than those that are breastfed. This is often due to the prebiotics that are present in breast milk that are quite unique and important. Preterms have a different microbiome than full terms. Again, an important factor in pediatric medicine. Genetics, while I did poo-poo the role of genetics in medicine, pun intended, a while ago, we now know that host genetics is a very important factor for heritability of microbial factors, and about 40% of the microbiome is heritable. So that's an important point as well, and we're getting to know more about that. I mentioned already about maternal stress, but also maternal infection, and that's been studied often in the context of neurodevelopmental disorders. Where you live, the environment you live in will influence your microbiome. And the microbiome of the built environment is becoming a really hot area where architecture and design and engineering are interfacing with microbiology to create an environment where we're exposed to in different ways to different bacteria. And even this is now also coming into the mental health literature. Recent work from Chris Lowry's group, putting forward the concept of the built environment and its importance for mental health relating it to the microbiome around us. Antibiotics are a key factor that will also influence the colonization early in life and have potentially long-term effects thereafter. A number of years ago, my colleagues at the APC Microbiome Institute in Cork published this paper in Nature, which is really interesting because what it does, it looks at the microbiome of the elderly. So a lot of the emphasis on microbiome research is focused on early life. Well, now this was in the elderly, and they showed, following up almost 200 individuals, that their health outcome, as some of them transitioned from living in the community into nursing homes, etc., correlated very well with their microbial diversity. And the more diverse their microbiome, the more better their frailty indices, their immune function, and crude cognitive measures. But that wasn't the whole story, and an interesting one as it was. But they went one step further and looked at what is driving the diversity of the microbiome. And what it was what they were eating. And the diversity of the diet would give a much more diverse microbiome. And while this is focusing on the elderly, and it's important demographic to be looking at in this regard, it also is important, has implications for all of us. Because unlike our genome, our microbiome is modifiable to an extent. And so diet is an important factor there. It also helped to, really contextualize when we look at the elderly. The brain-gut axis is very important to be studied. And as I said, I'm interested in stress, and I'm particularly interested in what chronic stress does to the brain, but specifically in relation to the elderly, we know that there's an inflammaging process ongoing already, and that stress will add fuel to that fire. And the way that the microbiome gut-brain axis is positioned, and we've positioned it central in this schematic here, it allows it to be a very good conduit for the negative effects of both stress and aging. But potentially through diet or other means, we could actually tinker with it to reverse some of these deleterious effects and have an impact on cognitive impairment, anxiety, geriatric depression, as well as social isolation, which are all very important factors in the elderly brain health perspective. Moreover, Science magazine before Christmas had a whole special issue on why we age, and they also had a whole section on the microbiome and aging from my colleagues in Cork. So it's becoming now more accepted that when we're looking at aging, that we can be looking at the microbiome as a way forward. And it's a whole new area in biology and medicine, that the microbiome could help us deal with some of the effects of aging and be the secretive aspects of aging, and the diet is important, except it's not new at all. This guy, Eli Metchnikoff, who won the Nobel Prize in 1908 for his seminal immunology work on phagocytosis and macrophages, was working in the Pasteur Institute in Paris. And Metchnikoff, later in his career, which is often the case, he came up with some out there crazy idea that were completely tangential to his previous work, where he was intrigued by why people in certain parts of Eastern Europe live longer. And he noticed and put it down to the fact that they ate a lot of fermented food, and particularly yogurts and that contained lots of lactobacilli. And he's written about it in The Prolongation of Life. And this was never really taken up by the mainstream science or medicine at that time, but it's intriguing that we're beginning to come around. If you look at what's happening in the whole human genome area right now, this guy, Craig Ventnor, has set up a commercial entity called Human Longevity, which is sequencing the human genome of the elderly. But they're not doing that in isolation. They're looking at it in the context of the human elderly microbiome and human elderly metabolome. And so perhaps, it's coming full circle in terms of what Metchnikoff put forward and gives us some insight. we're interested in this a lot ourselves, and so this is some unpublished data, but I thought I would share with you where we're going with this. We've characterized elderly mice and shown that they have all of these behaviors that I mentioned: increased anxiety, cognitive dysfunction, and we've been looking at what could be driving aspects of that. And indeed, when we look at their pro-inflammatory phenotype, they have an exaggerated baseline pro-inflammatory phenotype, and when we stress them, it's even further exaggerated. But also we began to start looking at the microbiome in these elderly mice, and again we do find, you won't be able to see all the details, but it's mainly the concept that changes in the microbiome could be driving the changes in the inflammatory phenotype, which could be driving the behavioral changes. And so this is something that we are quite interested in moving forward. And the literature is becoming more and more exciting in this. There's a very exciting paper that's been published online at the end of last year in a preprint scenario showing that Alzheimer's disease pathology doesn't emerge in animals that lack microbiome, and also in the immune system, aging is very important in this regard. So far I've talked about early life and the microbiome and stress, and aging and the microbiome and stress, and the other area that we feel deserves attention is the teenage brain and the teenage body in this regard. And looking at adolescent brain vulnerability is very important because it's an area where we can actually tap into the impact of diet and other changes that are going on. And we know that this is a time period when certain disorders are ripe to emerge, be it substance use disorders, schizophrenia, especially in males, as well as certain mood disorders and eating disorders. So, and this is something that we feel deserves a lot of more attention. And so we fed mice antibiotics throughout their teenage years, or months, weeks in this case, because it's mice. And we found that there was an anxiety phenotype as well as a cognitive phenotype that emerged, and that this was much more prominent in males than females for reasons that we don't understand. But we are seeing these sex differences time and time again. And I'll come back to that a little later. So, but all of this really gets us thinking about, well, what are the factors that are signaling from the microbiome to the brain, and how are they getting there? And I think that's an important area which we're only hitting the tip of the iceberg. We know that barrier function is very important. We know that the bacteria can affect signals through the vagus nerve. We know that they have direct effects on the immune system, but and it's also worth noting that bacteria are little factories for producing metabolites, be them neurotransmitters or short-chain fatty acids, for example. And here's just some examples. Work from Cork has been able to identify GABA-producing bacteria and bacteria that modify the composition of fatty acids in the brain. Work from Elaine Chow's, who's now at UCLA, has really shown how much certain bacteria can influence the synthesis of serotonin in the host. And Mark Lice, who's now in Iowa, has really put forward this whole concept that bacteria are delivery vehicles for a variety of neuroactive compounds, and we're only scratching the surface of what they may be. I mentioned barrier function because if your barrier becomes somewhat leaky, then it allows infiltration of a variety of immune cells, and that can have direct effect on the brain through stimulation by bacteria or bacterial byproducts themselves. And it's worth pointing out that the microbiome is an important epigenetic modifier. And some of the metabolites from bacteria, and short-chain fatty acids are probably the most studied here, can act as have direct effects on histones and be histone deacetylase inhibitors, for example. And we put forward this concept a number of years ago that really positions the microbiome in a way to fulfill many of the criteria of an epigenetic modifier. And of course, with transgenerational inheritance being so important of an area in biological psychiatry today, and a lot of long way to go to fully understand it, but bacteria are perhaps one of the best ways of vertical transmission from mom to offspring overall. We know that they will help, microbiome will help shape gene by environment interactions, and that's developmentally regulated, and it is potentially reversible, so fulfilling a lot of what we would see. So again, this is work that we're doing in Cork, and we're very excited about with Roman Stilling, a postdoc in the group, but it's we have a long way to go to really tease it apart. So how do we study this? That's one of the questions. How can you study this microbiome to brain signaling? What are the methods? Well, infection studies have laid the groundwork a lot of the way from the classical psychoneuroimmunology days. Moreover, germ-free studies I'll talk about. This is bacteria without any colonization in specific mice. Probiotic studies, where you give a specific strain that may have positive effects or antibiotic effects, are also very useful for delineating how microbiota can influence the brain. And lastly, fecal microbiota transplantation, taking someone else's poo. And I will come back to that before the end, I promise. So we fed animals antibiotics early in life in a very nice study that Siobhan O'Mahony did a couple of years ago. And what we found was that when these animals grew up, the good news for parents, and I have young kids who get antibiotics, is that there was no major change in cognition, anxiety, distress response, or immune response. But what we did find was that there was a visceral hypersensitivity, an increase in abdominal pain that was persisted into adulthood and persisted across the different antibiotic cocktails that we were investigating. And what's intriguing here also is that if you look at the microbiome of these individual rats at the time when we look at their pain responses, the microbiome is no longer different. It had resolved. But it tells us that the microbiome being changed early in life changed the wiring of some of the pain pathways, and then the microbiome went back to normal, but the pain pathways didn't, and so that this persisted. It also may have implications for infantile colic and other abdominal changes, but it's quite intriguing, and it's in line with the theories put forward by Marty Blazer in NYU, where he has really pioneered this concept that exposure to low doses of antibiotics early in life can have long-lasting effects on metabolism in general, and that we as a society are missing microbes that our ancestors had that would protect us from developing some of the Western disorders, be it obesity or multiple sclerosis or inflammatory bowel disease, for example. So again, a lot of cultural work is being going on between different parts of the world, looking at the microbiome and showing it's much more diverse in hunter-gatherers in Tanzania, for example, compared to Western societies. And so that's something that we need to really understand from a public health perspective. Germ-free animals I mentioned, and the idea of being germ-free has taken our attention for almost a century. Here is a sci-fi magazine from the 1920s, and you have a germ-free man. And then later in life, later on, the advent of germ-free mice and germ-free rats occurred, and this allowed us to have a really unique tool that will enable us to answer a very specific question. And that's all they can do is the microbiome involved in this process, yes or no? And there's a developmental aspect to that. There's no human correlate, perhaps only maybe the boy in the bubble, but these are the mice in the bubble. And growing up in a bubble, and this is a recent review we have that Pauline Musinski put together, trying to look at all the information we have from germ-free animals. And while their clinical translation is important, they have allowed us to answer these very fundamental questions. And I'll share with you now some of the data that has emerged on it and how important it has been. So at the beginning of this talk, I took you on the journey that we had in showing that stress in early life impacted the microbiome. Well, there was also really nice work from Japan, from Sudo's group in Japan, which showed that if you were born germ-free, if you didn't have microbiome, then you will have an exaggerated HPA axis to stress in terms of the ACTH or corticosterone response. And intriguingly, they showed that they could reverse this by putting in a single bacteria, bifidobacteria. They could reverse this. And this paper is very important to the field. And it took a while for people to really catch on to it and really appreciate the significance that it really has. And like is often the case in science, when you come up with an idea, many other groups also have the same idea and often publish before you in this regard. And this paper led people to start thinking, well, the neural circuits that are underlying the stress response should also be different then. And really important paper from Marcellus Heitz's group with Sven Pedersen in Karolinska, along with the paper from Jane Foster and Karen Ann Neufeld at McMaster, and then later our work, all showed the same thing in principle, that bacteria in the gut played a crucial role in mediating stress responses, anxiety-related behavior, and gene networks that were implicated in different complex behaviors, including anxiety. Here is some of our data that Jared Clark in the lab put together, which was to show that there was a decrease in brain-derived neurotrophic factor in the hippocampus, coupled with an increase in serotonin. And so this really tells us, you know, that the using germ-free animals, that we can really move forward and begin to interrogate this. It also is in line with really exciting work that was emerging at the time from Chemek Berchik and Steve Collins' group in McMaster, looking at the impact of antibiotics on BDNF levels in the brain also. More recently, we also focused on the hippocampus, but we started looking at other factors. And so we looked at adult hippocampal neurogenesis. And we know that we have continuously new neurons being born in our brain, and that these are sensitive to being to negative impacts of stress and inflammation and aging, and positive effects of learning and physical exercise, as well as antidepressant drugs. And what we found was, when we looked at what happens in these germ-free animals, we found that there is this change in neurogenesis in these animals, and indeed there was an increased neurogenesis. So the regulation of neurogenesis, the brake on it, is gone out of kilter in these germ-free mice. More recently, we've turned our way, again, this is unpublished data, but ones that we're very excited about, to the amygdala. And in the amygdala, we find that there's increased volume, increased spine density. When we look at the hypertrophy, there is a change in the size and the morphology of the amygdala. And this is work that Pauline Musinski has been doing for now some years in the lab to tease it apart. We've applied also a RNA-seq approach to look at what genes are being changed in the amygdala. We just published that last year. This is work from Roman Stilling and Alan Hoban and others in the group, and really showing that there are differential gene expressions. We took the same approach to another brain area, the prefrontal cortex, which is very important for higher cognitive and decision making, and also for regulating the fear and impulsive behavior. And And what we found was that when we looked at what genes were being changed in these germ-free mice, we found that there was a significant number of genes relevant to myelin, the insulation on nerve cells. And we validated this, that it was specific to the prefrontal cortex only, none of the other regions. And this is an area which we know the myelination patterns in this area is sensitive. Work from Mount Sinai has shown that this area is particularly sensitive to stress and chronic stress on myelination. And so here we get the opposite, a hypermyelination in terms of upregulation of the genes involved. And when we looked at EM, there was this hypermyelination occurring. So telling us that fundamental processes in the brain are regulated by the microbiome. Other studies from other groups, from St. Pedersen's group, has shown that the blood-brain barrier, for example, its integrity is regulated by the microbiome, and that you could reverse this leaky blood-brain barrier by providing bacteria that secrete high amounts of short-chain fatty acids. So that's quite important as well. Very nice paper from Marco Prentz's group in Germany last summer really changed a lot of how basic neuroscientists start thinking about the microbiome when they showed that the activation state of the microglia in the brain are under the control of the microbiome. And then if you looked at germ-free or antibiotic-treated animals, that this changed and that their elongation patterns were very changed, and that they could reverse this by giving short-chain fatty acids. So very intriguing, because we know that microglia are involved in so many processes relevant to aging, to synaptic pruning, and to neurodegenerative disease and the neuroinflammatory process. And now we have a way to regulate it through the gut. And that's very intriguing. It's also, didn't lose my notice that the microbiome is very much regulating something that is coming from the work of Metchnikoff. Metchnikoff discovered the macrophages, and microglia are the brain's macrophages. So again, reinforcing how right he was. I mentioned sex differences, and we find consistently in our germ-free animals that a lot of the effects seem to be more prone and more robust in males than females. And so that led us to looking at some of the behaviors that are relevant to male-specific disorders, neurodevelopmental disorders like schizophrenia or autism spectrum disorders. And so because we know that autism has 4-to-1 male-female ratio. And so what we want to do is look at some of the core symptoms of autism, such as abnormal social interactions. And so we looked at these germ-free mice, and mice, like humans, if you give them the chance to spend time with an empty chamber or another mouse, would prefer to spend time with the other mouse, but not if they lack microbes. Mice, like some humans, are a little bit fickle. They'll prefer to spend time with a new playmate versus an old one, and again, not if they're germ-free. They had increased repetitive behaviors, like we also see in autism. So it's telling us that for normal social responses, we will need to have an appropriate microbiome in our gut. And it's quite intriguing. It's in line with some of the interesting data that's emerging in animal models of two of the clinically known predisposing factors for autism, which is maternal infection or maternal exposure to valproate during pregnancy. And work from the Netherlands and from Caltech, from Sarkis Mazmani and the late Paul Patterson and Elaine Hsiao have shown that there is changes in the microbiome in these animal models, and which has led Jack Gilbert and Rob Knight to quite provocatively and perhaps prematurely to put forward the concept that we could be developing down the road probiotics for autism and other neurodevelopmental disorders. But this led us to start thinking a little bit about, well, why on earth, from an evolutionary perspective, would we require bacteria in the lumen of our gut to mount normal social behavior? And we start constructing, we now, know for a long time that elaboration of cortical function and brain maturity over throughout evolution has responded to being in social groups, and that we know that the microbiome is now playing a role in social behavior. And so we're beginning to close these loops in. And together with Seth Bordenstein in Vanderbilt, we started asking, could this play a role evolutionary in our social responses? And what Seth would always say to me is, always remember that the bacteria were there first, and that we have co-evolved and our brain has co-evolved with these bacteria. So this reliance on bacteria has been very important for our own brain development. And that bacteria and the microbiome are our friends with social benefits. So we're beginning to develop a construct where we can, as neuroscientists, we're intrigued by what changes in the brain occurs throughout our lifespan, how that may lead to the predisposition of certain brain and disorders. But now we must juxtapose it with what's going on in the microbiome at that point, and even in points earlier, and that's going to be very important. And so we're building a framework to be able to study this quite well. And as I mentioned, that stress, especially in early life, will change the microbiome, and that the mice lacking the microbiome have exaggerated stress response. It leads us to start thinking, could we modulate the stress-related brain health with the tinkering with the microbiome itself through probiotics? And in a study that Javier Bravo did some years ago, we looked at whether if we fed normal, healthy mice a lactobacillus, would it change their behavior? And indeed it did. They had reduced anxiety, reduced antidepressant-related behavior in the 4-swim test. Their corticosterone response to stress was reduced. And when we looked in their brains, there was wide-scale changes in GABA neurotransmission in their brains, which was quite striking for us. But again, the mechanisms for this needed to be resolved and are quite important to tease apart. And I've shown you this before. This is some of the communication pathways, and I did point out the vagus nerve. And together with John Bienenstock's group and Paul Forsyth at McMaster University in Canada, we performed a subdiaphragmatic vagotomy and cut off this signaling pathway. And lo and behold, all of the effects of this individual bacteria were gone. So as I like to say, this tells us that what happens in vagus will not just stay in vagus, but affect our whole emotions overall. And while this is just one pathway and not the only one, it's very important to try and delineate vagal, both bidirectional vagal interactions in mediating the gut-brain responses, and that we need to be able to come up with more rational ways of moving forward with this into translating into human studies, and as well as some of the side effects of certain drugs. So I mentioned fecal microbiota transplantation. The press love it. It's something that has that yuck factor. This is basically taking someone else's fecal material and it being delivered as a medicine. And if you had Clostridium difficile, C. diff, you definitely may want to try this if you were being resistant to treatment. And it has saved countless lives because these people are hospitalized and are slowly dying because they're not responding to conventional treatments. But it has, then now people are investigating it in other domains, in Crohn's disease, in metabolic syndrome, etc. But what about in psychiatry? And to my knowledge, there's no human studies published out there yet, but there's this very intriguing paper from Chemic Burchick and Steve Collins in McMaster, where they looked at 2 different mouse strains with 2 different bacteria populations, and these mice differ in their anxiety behavior, and they did a transplant, and lo and behold, they turned the resilient innate Swiss mouse into a valve C mouse, and vice versa. And so this also, if it has implications, if you move forward into human trials, that if you ever need to get a transplant for C. diff, you should make sure you have a very good psychological profile of your donor at the very least. So all of what I've told you really has been coming out of animal studies, and we can criticize animal studies because they have their limitations, of course, but they have really enriched this field and been able for us to move forward. And for some reason, there's been reluctance in clinical medicine to actually take this, and also industry has had problems grappling with how to enter into this because it's not a traditional pharmaceutical approach to treating brain health. But together with with Ted Dinan and Catherine Stanton, we've come up with this term psychobiotics, which are bacteria which were taken in adequate amounts will convey a positive mental health benefit. And this is something that we are now moving forward into human translation. And this is Andrew Allen, a postdoc in the group, testing a specific strain of bacteria that has emerged from our animal studies that we've shown to affect stress and cognitive performance in animals. And now we're moving it into humans and doing EEG and other laboratory stress tests. And this is proving to be really important and a very exciting area, one that we're very, buoyed by. And it's really new, except of course it's not. And there's this paper from the British Journal of Psychiatry in 1910 on the treatment of melancholia by lactic acid bacteria. So it's always good to be reminded. And of course, brain imaging is going to help as we move forward. And there's a really nice paper from Emran Meyer and Kirsten Tillish in UCLA from a number of years ago to show that a fermented milk product containing 4 or so bacterial strains had direct effect on emotional networks within the brain and dampening down the response in an fMRI. So this is where the field is going, and we're slowly moving there towards more psychobiotics. And there are studies emerging all the time. They tend to be small, and then we need to really move forward and have larger scale studies. And we also need negative studies because, of course, only a very small fraction of the bacteria that are out there are going to have positive effects. And we have to understand what is causing these positive effects and where they're going. So in conclusion, I kind of, shared with you our perspective, where the microbiome influences the brain. And I started thinking of it more and more like a puppeteer and puppet. And you can tell I have a son who loves Pinocchio. And then Pinocchio had many adventures in his life, but it was sculpted by this puppeteer, Geppetto. And basically, we're beginning to see that the microbiome could be the brain's Geppetto and help to sculpt it in early life and be always there when it gets into a little bit of trouble along the way, and have a very important relationship overall. And we're beginning to appreciate how this is through our entire lifespan, and that in the 20th century, our whole goal was killing microbes, whereas now we realize how important the microbiome is. And that it reminds me a little bit of TV shows like Downton Abbey or Upstairs, Downstairs, where you have two populations living in the same house, and they require each other for survival, but that they go about ignoring each other. And it's only when something happens downstairs that the real drama occurs upstairs. So I'll leave you with this, and that your state of gut will markedly affect your state of mind. I hope you have a great St. Patrick's Day, and this is the work of a lot of people in Cork. I want to point out Ted Dinan, my very close clinical colleague, who's head of psychiatry, and a whole host of many other great people, our funding agencies, and some of the industry partners that we have. And thank you for your attention. Okay. And now I'm ready.
Thank you, John, and a happy St. Patrick's Day to you too. We'll get right to the questions. Click on the green Q&A button lower left and submit your question. We have a lot of them today. Our first, have you examined the epigenetic link from stress to gut microbes and changes in the microRNA messenger RNA balance?
It's a very important question, and we're very much buoyed by the recent work that's showing how there is a clear relationship between the microbiome and host microRNAs as well. So we have an ongoing program tackling that right now, but it's too early to have any definitive data.
Thanks, John. Our next question, does the overuse of antibiotics in dairy and beef farming affect the microbiome?
This is really what Marty Blazer's Marty Blazer's hypothesis is showing is that especially in the U.S. where there is a food chain exposure to antibiotics more than in other countries, that this could actually be a very important aspect of this. And indeed, but definitive proof is difficult to get. But in his studies, in his rodent studies, he proved that early life low dose penicillins can have long-term effects on metabolic health.
Great. Our next question. Instead of medication, can we rely on a personalized diet therapy to recover from a stressful brain?
That's a really good question, and one that it's almost too early right now, but a lot of people are beginning to look at that. And indeed, nutritional psychiatry is really growing as an area. People like Felicia Jacka in Australia and others are really pushing forward this concept that we might be able to, through diet. And once we know more about our personal microbiome, and what diets could be used to actually shape that and how that can change our response to stress, you could see potentially how it could be used, at least as an adjunctive aspect to other behavioral or pharmacological treatments.
Great. Our next question. It's been demonstrated that probiotics could decrease anxiety and depression levels. What's your opinion about the use of probiotics to modulate the pain in any population with a microbiota alteration?
Okay. So we have to be careful because a lot of the studies with probiotics have not been subjected to what rigor that we would be used to in clinical medicine. So what is called a probiotic by definition means a live bacteria with a proven health benefit. And most of what's available, particularly in the US, haven't had that rigor attached to them. So that's one of the problems with the probiotic field in general. And a lot of the groups, the scientific groups, are trying to really move this forward. And that's why we like to, by pushing science behind it, we're able to move forward. But the point about relation to pain is very important, and we know that probiotics and potential probiotics can modulate pain pathways. And in irritable bowel syndrome, there is probiotics that have proven, one that was developed from my colleagues in Cork, and pain is a key pathway that's gone awry in irritable bowel syndrome. So I think it's a really key area for testing in the future and identifying what specific probiotics would have beneficial in pain and other aspects of stress.
Okay, this is our last question that we do have time for. Can gut bacteria secrete factors that endogenous cells use as part of their normal physiology, i.e. short or medium chain fatty acids?
They make a lot of yes, they make a lot of short chain fatty acids, butyrate, acetate, and propionate, and they're used in all aspects of host physiology and health, and are really one of the important regulators of microbiome to brain signaling, potentially.
Thank you so much, John. We've had so many great questions. We're out of time for that, but I'd just like to throw it back to John Cryan for any final comments for our attendees today.
I thank you for your tuning in live or on demand. This is a growing field. It's a very exciting field. It's good for neuroscience to think below the neck sometimes, and as you enjoy St. Patrick's Day, think about your microbiome and think about ways to keep it well, and all the best.
Thank you again, John. All the best to you too. Today's webcast will be available for on-demand viewing through September of 2016. You'll be receiving an email from LabRoots alerting you as to when this broadcast is available for replay. We invite you to share that email with your colleagues who may have missed today's live event. Thanks for logging on and participating. We hope to see you next time. Goodbye.